a brief introduction. What kind of data are we dealing with? Well, we use mass spectrometry to analyze proteomics samples. And so mass spectrometry, you can consider it a little bit as follows. So here, each of these Lego structures correspond to individual molecules. And so we want to figure out which molecules these are. For this, we use a technique, analytical technique called mass spectrometry, which measures the mass of each individual molecule very accurately. You can consider it as a super sensitive kind of scale. And based on the mass, we determine which molecule it was. However, this doesn't give us the exact peptide identity, because, of course, you can have multiple amino acids, the same amino acids sequence, but in different orders. Same way you can have, this yellow block, it can be in this position, but it could also be swapped with this green block. And in that case, this molecule still has the same mass, but it has a different sequence identity. So we need to do an additional step. This is called tandem mass spectrometry, in which the molecules are first smashed into pieces, and then we will measure the mass of all of the fragments as well. And then based on those fragment masses, we can build up the entire molecule peptide through the masses of the prefix and the suffix subsequences. So this is conceptually, but what does it look like in practice? Well, this is a mass spectrum that we would obtain during a mass spec experiment. Typically, we get tens of thousands of such mass spectra from a single run. On the x-axis, we see the mass over the charge, and on the y-axis, the intensity. And so these masses are the very accurate masses of the fragments of this peptide that was smashed into pieces. Now, from this representation, we want to figure out which peptide was measured, so which peptide this spectrum corresponds to. If we have a certain indication that it could be, this peptide sequence here, then we can look for all of the different fragments in between each amino acid residue. B ions go left to right, or N-terminal to C-terminal. Y ions go right to left, or C-terminal to N-terminal. For each of the fragments, we can calculate its theoretical mass, and then we can check whether there are peaks in the spectrum that have the same mass. And here we see that we have very nice coverage of the full Y ions for this peptide, and several of the B ions as well. So we can be pretty confident that this is indeed the correct peptide for this mass spectrum. And so that is more or less what traditional approaches in mass spectrometry-based proteomics do. So we have our biological sample, we extract the proteins from it, we digest those proteins into peptides, and then for each peptide, for the various peptides, we acquire tandem mass spectra. At the same time, we have our theoretical protein database. We convert those protein sequences into peptides as well. And for each peptide, we simulate theoretical spectra because we know at which locations the B and the Y ions should occur. Then we take our experimental spectra, we match those to the theoretical spectra, and based on which spectra match well, we then assign the corresponding peptide sequence to our experimental spectrum with a certain score. And this is the traditional sequence database searching strategy, in which we have a reference proteome in our sequence database that we can search against. However, in some cases, we might not have such a reference proteome. Does that mean that we are completely out of luck, that we can't do anything at all with our mass spec data anymore? Well, luckily, no. We can use a technique called de novo peptide sequencing, where we derive the corresponding peptide sequence directly from the mass spectra without any external additional information. So no reference proteome, no candidate peptides or theoretical spectra are used. Just by looking at the distances between different peaks, whether they correspond to certain amino acids, delta masses, etc., enables us to reconstruct the peptide sequence directly from the mass spectrum. And a few applications in which this is especially relevant is, for example, immunopeptidomics, where we don't have standard peptides, but basically any protein subsequence can be a peptide because they are generated by the immune system. Metaproteomics, where we're searching not against a single organism, but against hundreds or thousands of organisms. And so we would have to deal with a very large search space that is challenging for standard tools. Paleoproteomics is another example where people might study historical samples for which no reference proteomes are available. And for example, venomics, where we're studying the venom of snakes, which are also for which no reference proteome is available because there's a lot of mutations in those peptide and protein sequences that are not covered in existing databases. So these are some of the typical examples where de novo peptide sequencing is used. But even for standard proteomics applications, it can be relevant to make new discoveries because you're not constrained by a database search, by a database anymore. And so how did we represent de novo peptide sequencing? Well, we represented this as a translation problem, where rather than from translating from one language to another language, here we translate from the language of mass spectra, which are represented by sets of peaks, to the language of peptides, which are represented as sequences of amino acids. And by positioning this problem in such a way, we can use recent advances in deep learning and large language models and apply these to the de novo peptide sequencing problem. And that's exactly what we did. Our tool is called Casanovo. So this is a transformer-based de novo peptide sequencing model. If you're familiar with transformers, you'll notice that the diagram on the right-hand side pretty closely matches the typical definition of a transformer encoder-decoder model, where we have as input to the encoder a tandem mass spectrum, where each peak is represented by a mass and intensity. The peaks are subsequently encoded using a transformer positional encoding system, and then processed by a transformer encoder to learn a latent representation of the entire spectrum that is then fed to the transformer decoder. The decoder gets some additional information from the precursor, so the precursor mass and charge, and then autoregressively predicts amino acids. So this means that it will predict each amino acid at a time. So in this case, it will first predict the P. So it will first get like a start decoding instruction. The first amino acid that it will predict is the P. This will be fed back into the decoder to then predict the next amino acid, which will be an E, which will be fed back into the decoder, etc. And then finally, there's also a beam search decoding step to maximize the score of the entire peptide sequence. And so we introduced this Casanovo transformer-based model. And then
Professor,
yes?
on the previous slide, yes, on the first input layer, I see we have the mass spectrum as the input, the precursor, and then I think the previous sequence. So we have looked at the spectrum while you were starting this discussion, but I'm missing the translation to sort of like how to encode this input. How is it input like to have these sequences of or like token like inputs, the M naught, I naught, into the peak encoder, but also the precursor, like what differentiates it from these other tokens? I'm seeking some more clarification and light on that input layer, so to say.
Yeah. So the encoder receives just the peaks of the spectrum as input. And so each peak is represented by a tuple of two values, so the mass and the intensity. And those are both encoded using a sinusoidal representation. Rather than providing them as individual floating point values, which does still work, but is typically a little bit less expressive for the neural network. We use kind of the typical sinusoidal encoding approach, as in the original transformer paper, to represent these values in a little bit more granularity. And so that is then used for both the peaks in a spectrum as well as for the precursor mass and charge. Does that make sense?
Yes, but also because somewhere you've talked about like the process that is done in natural language processing, where you have these sequence tokens and you have the start token and the end token, and I'm trying to now link the two together from this tuple of peak representations and that sequence of the tokens with the start and the end. Maybe I'm missing something.
Yeah. So the encoder always gets the fixed number of tokens as input. So this is maximum 100 peaks. If the spectrum has fewer peaks, then there's just some zero padding. And that then learns a latent representation, right? So this is just the like a 768 dimensional representation of our mass spectrum that was used as input. This is then provided as input to the decoder, as well as this precursor information that is kind of used as a bit of the start token, this precursor information. This kind of tells the decoder to start decoding, and then it will autoregressively predict the output peptide sequence until it also then predicts a stop token, which indicates that the full peptide sequence has been produced. So it's basically exactly the same as in a typical transformer decoder.
Yeah, sure. Thank you. Just intuitive and it's a little bit ingenious. Thank you very much.
Yeah. And so we developed Casanovo, and then we compared it with a few other tools as well. And so here, what you see on the right, this is a peptide precision versus coverage plot. So on the x-axis is the coverage, so the number of or the fraction of spectra in a data set that we make predictions for, and then on the y-axis is the peptide precision. So a perfect tool would be here in the upper right-hand corner, right? Making correct predictions for all of the spectra in our database. We have Casanovo. We compare to Deep Novo, which was an earlier deep learning-based de novo tool using a combination of CNNs and RNNs. And then Novor, which was a traditional machine learning-based approach using random forests. When we trained Casanovo on the same data set as Deep Novo, consisting of approximately 1 million spectra, we see that it already outperformed both Deep Novo and Novor quite nicely. But then a benefit of the transformer architecture is that it really scales very nicely and very well if you give it more data. So when we increase our training data set from 1 million spectra to 30 million spectra, now we see a very strong boost in the performance, and basically on this subset getting very strong performance with an average precision of 0.95. So this is one of the benefits of these transformer neural networks, is that they scale really well if you give them more training data, and kind of set a new state of the art in the field, outperforming all of the previous methods. One thing, though, one setting in which it performed less well initially is that the model exhibited a tryptic bias. So most proteomics data is generated using trypsin as the protease, which ends in arginine or lysine, those peptides. So after arginine and lysine, trypsin will cleave. And Casanovo was trained almost exclusively on such tryptic peptides. However, if we then applied it to immunopeptidomics data that don't follow these tryptic cleavage rules, but that can basically end in any C-terminal amino acids, we saw that performance dropped quite significantly. And that is mainly because it was still like over predicting peptides ending in arginine and lysine, whereas peptides ending in different amino acids were underpredicted. So this, on the right-hand side, this is a plot of the C-terminal amino acids for these aminopeptides. So we had to adapt our training mechanism a little bit and build a more balanced training data set. And you can see that now in blue, that if we had a more balanced training data set, our performance goes up, and the bias towards certain C-terminal amino acid residues also disappeared. You'll still notice that this performance is lower than on the previous slide, but that is in large part because the data set immunopeptidomics is simply more challenging to figure out these peptides, and this is true for any methods, not only de novo sequencing. So if we then looked at whether we can make new discoveries with Casanovo, we use an immunopeptidomics data set again, and then we compared Casanovo against kind of the gold standard performance from sequence database searching using the TIDE search engine. So TIDE kind of got here about 1,600 peptides that unique immunopeptides that it could identify. If we looked at Casanovo results that were also in the TIDE results, we see that Casanovo was able to reproduce quite a few of those. But then if we also looked at Casanovo results in the TIDE results without any false discovery rate control, or Casanovo in the human proteome, we saw that we now identified up to 65% more unique immunopeptides. And one challenge of de novo peptide sequencing is that it doesn't really have established false discovery rate techniques. But considering that de novo sequencing can predict any possible peptide sequence, right? There is no reason why it should predict a peptide sequence that is in the human proteome. So if it does predict a peptide sequence that matches the human proteome, that is a strong indication that it is a correct prediction. And so here we see indeed that, by matching to the human proteome, we get up to 2/3 more identifications compared to database searching gold standard. We also used a tool called NetMHCPan, which can take an immunopeptide sequence and then predict the binding affinity to a known HLA types. And we did this for database searching results with TIDE and the Casanovo results. We see that immunopeptides predicted by both Casanovo and TIDE, as well as those by Casanovo only, show a very high binding affinity. And here this dashed line is kind of, this indicates strong binders. So 80% of those predictions can be considered strong binders, whereas the peptides that were predicted by the database search only were considered to be much weaker binders. So this kind of is a bit of an orthogonal evaluation that indicates that the peptides that Casanovo was able to find, including those that are unique to Casanovo that were not found by TIDE, are very plausible candidates for these known HLA types. Here's a result from a completely independent group. So this is another research group that took our Casanovo model and then applied this to a different problem, namely antibody sequencing, where also antibodies have extreme sequence heterogeneity. You can't really do this with database searching, and you need de novo sequencing. And they compared Casanovo to a few other models, and you can see here that across the various settings, Casanovo was always giving the best results. So kind of independent validation of the strong performance provided by Casanovo. So that is Casanovo in sequence database searching mode. We then considered, most people still, in de novo searching mode, but most people actually still use sequence database searching because, after all, if you know which proteome to match against, that's very strong information to filter your search space. So can Casanovo help us there as well? Not in its initial way, but maybe we can use Casanovo as kind of a score engine to measure how well our peptides and experimental spectra match to each other. And so we use Casanovo in kind of a teacher forcing mode to provide it with candidate peptide sequences and then get scores for peptide spectrum matches against our experimental spectra. And we compared this to 3 different search engines that are commonly used in proteomics: X-score, the HyperScore, and the Andromeda score. And here you see on the y-axis the number of peptides that were identified for increasing false discovery rates. And in blue, we see that Casanovo significantly outperforms all of the 3 traditional database search modes, which are, manually defined kind of heuristics. On the right-hand side, in the upset plot, we also see that Casanovo confirms the peptide identifications from the other search engines, overlapping with all of these different search engines, but then also has a very large proportion of unique peptides that I was able to find. So very interestingly, by first training Casanovo in the de novo sequencing task, we could then actually repurpose this trained transformer model and use it for sequence database searching as well, and get very sensitive PSM scoring. And then in our most recent paper published earlier this year, we released Casanovo version 5, whereas the previous results were from the last year's model version 4. In version 4, so Casanovo, it will provide amino acid scores because it makes predictions for each amino acid at a time. And so those amino acid scores can be considered the conditional probability of each amino acid token being correct, given the prefix peptide sequence. And so then in version 4, to get to a peptide score, we calculated the arithmetic mean of those amino acid scores. An important downside of this is that this score is not really calibrated. And what does that mean? Well, here on the x-axis, we see the Casanovo score, and on the y-axis, the probability of a peptide being correct. And you can see that even for very high Casanovo scores in the range 0.8 to 0.9, the probability of the peptide being correct drops very quickly. So as a consequence, people would need to set really high thresholds, like 0.999 even, to try to avoid false positives. In version 5, we started using the product of the amino acid scores as a peptide score instead. And this gives a much better calibrated score, where you now see that the peptide score kind of very nicely correlates with the probability of the peptide being correct, and maybe even here in the being a little bit conservative in the high score region. But this is very beneficial for users, so that now the peptide score has much easier to interpret semantics in that if you set a score of 0.8, about 80% of your predictions will be correct. Another benefit of our latest version 5 has
Professor,
been, yep.
Professor, on the previous slide, yes, it looks like when you look at the vertical axis from this slide and the previous slide, there has been a change in the values on the vertical axis. Actually, yes, I'm seeing the product and the arithmetic mean. So I'm getting the feeling like there is a multiplication happening, and when we multiply the probabilities, it looks like the performance is improving.
Yes.
And I'm wondering, how is this product introduced of multiplication that leads to the better performance?
Yeah, so this is done as a post hoc calculation to derive peptide scores from the amino acid scores that are produced by the model. So this is just first making a prediction, and then either initially we took the mean of those amino acid scores, and now in the latest version we take the product of those amino acid scores to give a single peptide score.
Okay, great. So this multiplying like the amino acid scores or probabilities sort of like gives better calibration, so to say.
Exactly. And this also makes sense if you think about the amino acid scores as conditional probabilities for each residue given its prefix. And so, you can multiply those conditional probabilities to get the probability of the full peptide sequence. So semantically, it also makes more sense.
Okay, thank you.
Right. And then another nice improvement in version 5 is that we put quite some emphasis on runtime performance. So in training between version 3 and between version 4 and version 5, training became 11 times faster, and inference afterwards became 3.5 times faster. So making it a lot more efficient. That said, you'll still want to use a GPU to run it on real data sets. If you run it on CPU only, it's still quite slow. And database search is also significantly slower than de novo search because in database search, we have to evaluate dozens to hundreds to sometimes even thousands of candidate peptides per spectrum. Whereas in de novo searching, you only have to make a single prediction per spectrum. And so to wrap up this part about Casanovo, we use kind of the latest advances in deep learning and transformer neural networks to position de novo sequencing as a sequence-to-sequence translation problem. Doing this, we get excellent performance across a wide range of applications, and we're continuously improving Casanovo for better performance, better accuracy, better runtime scalability, and so on. But of course, Casanovo is not the only tool that exists for de novo peptide sequencing. I've shown previously in the comparison and evaluation against Deep Novo, which was the first deep learning-based tool for de novo peptide sequencing, introduced in 2017. In 2020, there were a few more tools. Then in 2022 came Casanovo, as well as a few other tools. 2023, even more models building on Casanovo, using transformers now as well. 2024, people kept publishing new tools, etc. So there are now many different tools that people have introduced, and all of these individual papers, kind of claim that their tool is the best. Somehow that can't all be true, right? Even though some tools might be better in some use cases, there's no such thing as one universal best tool. But we need to understand in which use cases, where do the different tools work well? Where is de novo a good solution, or where does it still fall short? And so we need a kind of consistent, systematic benchmarking. And that's a project that is ongoing in my lab at the moment, where we have compiled a benchmarking data set from 29 different proteomics experiments, consisting of 24 million spectra in total. Those were analyzed with 3 different sequence database search engines, and we get kind of a gold standard of 6.4 million annotated spectra. The data sets come from various different species, so human, other model organisms, other plants, animals, metaproteomics, etc., processed with different enzymes, trypsin, other enzymes, immunopeptidomics, non-specific cleavage, and from different mass spectrometry vendors, to really try to cover the broad range of proteomics approaches that one might want to tackle with de novo sequencing as much as possible. And then importantly, the data was divided into public and private test data. So about half of the data is public test data. For these, the exact origin of the data sets and which public data sets on Pride or Massive they come from is available. But then there is also half of the data that is a private test data, for which only general characteristics of the spectra are provided, but the exact origin is not publicly communicated, to make sure that we maintain kind of an independent test set, and that people are not kind of overfitting to the benchmark by training their models on this data. We then collaborated with developers from 18 different de novo tools who contributed this to our benchmark by implementing it in an Apptainer container to run in our runtime environment. And with each of these 18 different tools, we make predictions for all of those benchmarking data sets. And then we compare those predicted PSMs to the ground truth PSMs, and we calculate metrics to evaluate the performance of these different de novo tools. We calculate peptide and amino acid level precision coverage, as in some of the previous slides I've shown. But then we also calculate database independent evaluation metrics, such as matches to the reference proteome as before, but also the difference in predicted fragment ion intensities and predicted retention times for those de novo PSMs. And then these results are shared in an online dashboard, where it's also all of the tools and the metrics are periodically rerun, so that it automatically tracks the state of the field as new versions of the tools are produced and released, and as new tools are added to the benchmark as well. And this is still an effort that is ongoing, but I can already give a few preliminary results. For example, we see here that when running the different de novo tools on data sets generated with different proteases, we see that for tryptic data, most tools, and Casanovo is highlighted in green, but then the other tools that are evaluated are highlighted in gray to not make the plots too complex. But we see that most tools actually work quite well for tryptic data. If we look at alternative proteases, performance tends to drop across the board. Whereas, for example, for immunopeptidomics, for non-tryptic cleavage, performance is still very good. And this is also in part because immunopeptides, HLA-1 immunopeptides, tend to be relatively short. So this is kind of the sweet spot for de novo peptide sequencing. We also see performance variances based on organism. So most tools, again, are trained on human data, and here performance is on average again the best. If we move to other organisms, performance can drop to a more or lesser extent. We do see maybe some challenges with analyzing different organisms. But on the other hand, as we see here for the fungus data set, we also don't see complete overfitting to the human training data either. And then the same thing for different instruments, where we see good performance on thermal Orbitrap on the left and SciEx swivel top instruments on the right. But then also for Bruker timsTOF, none of the tools perform very well. And this is in large part because of the ion mobility dimension that is included in timsTOF measurements, which none of the tools can accurately deal with yet. And then finally, an example of our database independent evaluation. And here we see on the left-hand side the difference between the predicted retention times and experimental retention times. The database searching results as kind of as a baseline are indicated here. And we see that the high confidence predictions tend to very closely match the database searching results, whereas for the lower confidence predictions, the retention time deviations become larger, which kind of indicates that you might use this retention time difference as an additional filtering criterion to select the high quality peptide identifications. Same for spectral angle between predicted fragment ion intensities and experimental spectra. For high confidence predictions, actually, the spectral angles are even better than for the database search, because this is exactly the region where if you have a strong fragment coverage, this is the region where de novo searching works well. Whereas for lower quality predictions, the spectral angle and the fragment coverage is lower, which also makes sense, of course, because there is just less information in the spectra than for the de novo tools to make correct predictions. And so, in conclusion, why do we need to do this benchmarking? Well, there are now dozens of different de novo tools, and we need objective evaluation. Not necessarily to say this is the best de novo tool that everyone should use, but rather to identify strengths and weaknesses across the tools and figure out in which settings de novo sequencing is a good option or where it might still struggle. And so based on our initial results, we do see indeed that these deep learning-based de novo sequencing tools tend to get very strong performance in well-defined applications, but performance can still drop in less common scenarios, such as instruments that were not represented in the training data set or different proteases, etc. It's also, this benchmark is also intended as a living resource that will be continuously updated with new data sets, new tools, etc., to kind of track the progress of the field as de novo searching tools become better and are able to address more and more scenarios. And in that way, it's a resource for both users and developers. For users to understand how de novo will function, which tool they might select, and for developers also to provide a comparison to other tools on a consistent benchmarking data set. And so for developers to contribute their tool, they just need to open a pull request with a containerized version of their tool, and then it will automatically be integrated in our benchmark. And so, this is my team at the University of Antwerp. These are students and postdocs that do all of the research. And if you're interested in Casanovo, feel free to check it out on GitHub. It is open source, as well as the various publications associated with Casanovo. And I'm happy to answer additional questions if you have any.
Thank you, Prof. Definitely. For me, the first question is about the mass spectral that is being sort of like, manipulated to form the input. And then somewhere, I hope I didn't misquote you. You're saying you're taking the peak values within the spectral. And I question, or I ask myself, like, isn't there data loss, and how does the transformer in this sense like compensate for that loss if it is there, to make the perfect prediction of the sequence? Like, how do you cater for that if at all it happens anyway?
Yeah, no, this is a good question, and it's actually an advantage or a strength of transformers compared to older deep learning approaches that, for example, used convolutional neural networks or just like fully connected neural networks. Because older approaches used to represent spectra by just binning across the mass range and then using 1 Dalton bins and then assigning the peak intensities to the corresponding bins. This has several disadvantages. 1 Dalton bins are pretty coarse, so you lose a lot of the high resolution mass information, but you can't make the bins smaller because then these vectors just become too large to still fit in your GPU memory. Another downside is that there can be edge effects. So there's always a little bit of uncertainty associated with the mass measurement. So if it falls right on the edge of a bin, it might also have been just in the neighboring bin. And so this is a downside of these binning approaches and convolutional neural networks, where in contrast in the transformer, we just provide the exact mass and intensity value directly to kind of the peak encoder, which then uses the sinusoidal encodings to represent these values. So we provide the masses at their native resolution to the model. So we avoid kind of these artifacts due to binning.
Okay, that's great. Thank you. I think my next question is somehow interlinks to beam search. But fundamentally, it is about the performance improvement you talked about of 11 point something, which is really overwhelming. And for me, I want to know the engineering fit there, whether it was about speeding up that actually comes from data loading, or the GPU compute, or the beam search itself?
All three, actually. So data loading indeed was a big factor, especially during training, where first we were loading kind of the spectra from MZML files or MGF files on the fly. And then in version 5, we kind of used a LANS based format as a backend. So LANS is kind of a columnar data format that can that provides very efficient I/O speeds. So that was a big factor. The GPU efficiency was also a big factor. So we put some serious software engineering effort into trying to avoid Python. First we were doing like some for loops in Python, which was very slow, and then of course we tried to move as many calculations as we could directly on the GPU to do it there. And then also the beam search. The beam search was actually one of the most. It's a complex piece of code because Beam search, inevitably, you do need to do some steps in Python, and that's where there were definitely some inefficiencies. And that is then where the kind of threefold speed up during inference comes from to a large extent. So it was really, trying to look at doing first runtime profiling, checking where the bottlenecks are in the code, and then systematically trying to address those issues, and then moving on to the next big bottleneck, and so on.
Thank you. Thank you very much, Prof. I think I've gotten one more question. In the meantime, you guys, if anyone has a question, just unmute yourself. Prof, the one question that I can't go without asking is about of course, we know the transformer has established itself and it turns out to be a powerful architecture. And so for the deliberate use of where you talked about using the sinusoidal encoding when you're looking at the spectral peaks, because we know that from the traditional transformer, I think they also somewhere use that sinusoidal encoding within the language. So did you just like look at the work that the authors of the transformer did, or it was really deliberate or intentional? Actually, they also used, I think, the cosine, but
Yeah.
you seem, I think, to have picked just the sinusoidal. And is there a reasoning for that?
Yeah, no, we tried various different things, and I think that this is actually often where the creativity and computational biology research is needed, right? Because most deep learning new architectures and models, etc., are first introduced on either images or natural language, or maybe speech or something, right? And that's what most benchmark or traditional training data sets consist of, which makes sense, of course, because we're all naturally familiar with images and text, and you don't need to study that first. Whereas here we're dealing with mass spectra. These are complex data types that AI researchers don't know how to interpret. And so it's always kind of or it can be tricky to adapt architectures from natural language to our specific setting in mass spectrometry. And so that's where kind of the creativity comes in, and trying to figure out how can we best encode our mass spectral data in such a way that we can use it with these kind of newly proposed architectures. And so we tried various different things, just like providing the values directly, or using various encoding ways of encoding the values, various binary encodings, etc. But then ultimately, empirically, the sinusoidal encodings give us the best performance. So that's then what we ended up using.
Wow, fantastic. Yeah, that's really some creativity. If there are no other questions, I want to thank Professor Wood for taking off his time and really share this wonderful work with our group. Thank you very much, Prof, for really taking off your time and decide to